Yeah, good morning. Topic of my presentation is on cancer immunotherapy. But before that, let me introduce myself. I am Dr. Neesar, Associate Professor, School of Life Sciences. I did my graduation in biochemistry honors in 2002 and master's in biotechnology from Aligarh Muslim University. After qualifying the CSER NET, I was selected for NCCS Pune to complete my PhD in 2009. Further, I went to Detroit, Michigan for a postdoc at Kamanath Cancer Institute. Okay, my content for the today's talk is cancer and the immunology, cancer gene, tumor antigen, cancer and the immune response, and cancer immunotherapy. Coming back to the topic cancer immunotherapy, to know more about this, we need to have a basic understanding of the cancer immunology, which consists of two components. One is the cancer and other is the immunology. So cancer, there are two categories, benign and the malignant, and the immunology, the immunity can be categorized into two, innate immunity and the adaptive immunity. So innate immunity is basically which is acquired by birth. It is a non-specific, which does not have any memory, and both the cellular and the soluble factors both are involved to provide this innate immunity. However, the adaptive immunity is the delayed one, and two categories are the humoral, that means the soluble factors like antibodies, and the cell mediated mainly by the lymphocytes. So there are two categories of the lymphocytes, the T helper cell and cytotoxic T cell. There are several subsets of the T cell that play a very important role in providing the adaptive immunity. Uh, Let me just compare about the normal cell and the cancer cell. So the normal cell actually, uh, they know when to start dividing and when to stop. So upon getting the appropriate stimulation, these cells will start dividing, and on getting the stop signal, they will stop dividing. However, the cancer cell, which is uncontrolled cell division, so the cells will continue to grow and divide, and you will see like there will be a variation in the size and the shape, and the nucleus will be larger than the normal cell, that will be darker than the normal cell, and an abnormal number of the chromosome, basically aneuploidy is seen, and which is totally disorganized in case of the cancer cell, and the cancer cell just keep on dividing and dividing, forming a clusters and without any boundary, and serve as no function. So cancer, the gene which is responsible to induce cancer is termed as the cancer gene, and they affect the growth of the cancer directly, and some of the gene they are involved to enable the metastasis also. An estimate of 300 different genes are involved in the human cancer, and that constitute of 1% of our total genome. So the two categories of the cancer gene is the oncogene and the tumor suppressor gene. So oncogene is basically the mutated form of the proto-oncogene. And the proto-oncogene, they are very much required, and they are the positive control of the cell growth. And we see there are 100 different types of the proto-oncogene, and these proto-oncogene generally they pose the growth factors or the protein kinases. And compared to the tumor suppressor gene, and these genes this is also known as the loss of function gene, and it reduces the probability that a cells in multicellular organism will turn into the tumor. Examples of the oncogene is the MYC and the RAS, and the tumor suppressor gene, you can say like P53, which is the guardian of the human genome, BRCA1 and BRCA2, which is involved in the breast cancer. Now, talking about the tumor antigen, so most of the tumor antigen give rise to the peptide, and that is recognized by the immune system following the presentation by self-antigen histocompatibility complex MSC molecules. In fact, many of these antigens have been identified by their ability to induce the proliferation of the antigen-specific cytotoxic T lymphocytes or the helper T cell. Basically, there are two categories of the tumor antigen. One that will express the self-peptide, and the second one which will be presenting the altered peptide. So the cell peptide is a result of the overexpression. So when there is a overexpression of the novel protein, so that constitute the tumor associated antigen, and the one which is specific, which is the altered protein, will present the peptide by the MSC1 molecule. They come in the category of the tumor specific antigen. So basically, the two categories will be tumor specific antigen and tumor associated antigen. And tumor associated antigen is because of the overexpression of a particular protein, or the inappropriate expression of the embryonic gene. Now let us see the tumor evasion of the immune system. Uh, Cancer cell employ various strategies to escape the tumor, the immune response. Generally, the tumor cell, the frequency express low level of the class 1 MSC molecule. So, we have seen the role of this MSC class 1 molecule, which is very important in presenting the tumor antigen by the cytosolic processing of the antigen and presentation. So once the altered protein is degraded by the proteasome and then loaded on the MSC1 molecule and displayed on the surface, so this MSC1 peptide is recognized by the cytotoxic T cell, and as a result, the cytotoxic T cell is going to release the perforin enzyme and will target and kill the mutated cell. However, the cancer is so smart that it decreases the expression of the MSC1 molecule. Next, the tumor cell may provide a poor post-stimulatory signal. For example, the B7 that is present on the antigen-presenting cell and the CD28 present on the T cell. So the interaction is very important. Apart from the T cell receptor binding to the peptide displayed by the MSC1 molecule. So this co-stimulatory signal, this interaction is also important. So both the signals are needed to induce the IL-2 production and proliferation of the T cell. The T cell will receive only a partial activating signal which may lead to the clonal energy. So anti-tumor antibodies can enhance the tumor growth. This is something very surprising, and the antibody, in fact, that is helping the tumor growth, and this is due to the antibody binds to the tumor-specific antigen and masks the antigen from the cytotoxic T cell. Also, the complexes may inhibit ADCC, which is antibody-dependent cell cytotoxicity, by binding to the Fc receptor on the natural killer cell or macrophages and blocking their activities. The antibody basically modulate the tumor antigen. So you will see like a certain tumor-specific antigen have been observed to disappear from the surface of the tumor cell in the presence of the serum antibody, and once reappear when the antibody is no longer present, and this phenomena is something called as antigenic modulation. So there are three different stages of the cancer editing. The first one is the immunosurveillance, which is called the elimination. Here the immune cell will be in the surveillance, and wherever they find the mutated cell, the peptide, the altered peptide displayed by the mutated cell, they are going to target that, and the cancer is eliminated. However, in the phase 2, there is a equilibrium phase, which is a fight going between the cancer cell and the immune cell, and there is a equilibrium. So there is this results in the genetic instability, and as a result, the cancer manipulates the environment and suppresses the immune system, and thereby resulting in the cancer progression. So that phase is called the escape phase. So basically, the three E's are important in that helps the cancer cell to survive and escape the immune response. So basically, in the elimination phase, we will see the anti-tumor microenvironment, and with the high expression of the pro-inflammatory cytokines, while it gradually progresses towards the pro-tumor microenvironment with the high expression of the TGF-beta and IL-10. So considering these properties of how the cancer manipulate the immune system, so we have different strategies which can be used as a cancer immunotherapy. The first one is like manipulating the co-stimulatory signal that can enhance the immunity. For example, use of the transfected tumor cells for cancer immunotherapy. So the cancer cell is transfected with the B7 gene, and uh, so as a result of this, there is a combined signal. The cytotoxic T lymphocyte differentiate into the effector cytotoxic T lymphocyte. The progenitor will be differentiated into the effector cytotoxic T lymphocyte, which can mediate the tumor destruction. And the transfected cell will act as a strong antigen presenting cell. Second, the tumor cell will be transfected with the gene encoding for the GM-CSF, that is granulocyte monocyte coronary stimulating factor, that allow the tumor cell to secrete high level of the GM-CSF. And this cytokine will activate the dendritic cell in the vicinity of the tumor, enabling the dendritic cell to present the tumor antigen to both T helper cell and cytotoxic T cell. Enhancement of the APC activity can modulate the tumor immunity. Another way to expand the dendritic cell population is to culture the dendritic cell from the peripheral blood progenitor cell in the presence of GM-CSF, TNF-alpha, and IL-4. The cytokine therapy can augment an immune response to tumor. Among the cytokines that have been evaluated in the cancer immunotherapies are interferon gamma, IL-1, 2, IL-4, IL-5, IL-12, GM-CSF, and TNF-alpha. There are monoclonal antibodies that are very effective in treating some tumor cells. For example, the monoclonal antibody against HER2 receptor is very successful. Another very promising immunotherapy is the adaptive immunotherapy. This technique involves the isolating of the TILs, that is tumor infiltrating lymphocytes, primarily activated cytotoxic T lymphocytes with the anti-tumor reactivity found within the tumor, expanding their number artificially in the cell culture by means of human recombinant interleukin-2, and the TILs are then put back into the bloodstream along with the IL-2, where they can bind and destroy the tumor cells. So this is the diagrammatic representation, where initially from the patient the blood the isolated the dendritic cell, which is cultured and then stimulated with the tumor antigen, and once activated it is infused back to the patient, where it present the antigen to both the T helper cell and cytotoxic T cell, as a result in attacking the cancer cell. So that's all. Thank you. If you have any query, you can post on Neesar.sls@education. The references you can take the QA 7th edition, chapter 90. Thank you.